Hi everyone, my name is Amin. I'm a graduate student at Columbia University, and today I'm going to present my accepted paper in MICCAI 2020, which is titled Demixing Calcium Imaging Data in C. elegans via Deformable Non-Negative Matrix Factorization. So we know that the calcium indicators are used as a surrogate for the neuronal activity, and there's been a great advancement in the imaging techniques, and currently we can image neuronal populations deep within the brains of really moving animals. Therefore, the extraction of calcium signals is crucial towards understanding neural dynamics. However, it is a very challenging machine vision task because of the limitations of imaging modalities and the spatial overlaps that the components will have in the videos. The problem becomes even harder for the C. elegans. To give you a quick background on C. elegans is a worm. It's a transparent nematode that has a very simple nervous system structure, and for this reason, it is well studied in the field of neuroscience. Some of the factors that contribute to the difficulty of the problem for C. elegans is that the data sets that we deal with are relatively large. So they're 4D or 5D tensors or volumetric images in multiple color channels. Videos have motion. So the worms are either semi-immobilized or they were moving. In both cases, they have both motion, and the motion is not linear. There's also limitations in the spatial or temporal resolution of the videos. So we cannot have both perfect spatial and temporal resolution because of the limitations in imaging modalities. And usually the cells overlap in the videos, and the neural signals are also correlated. So I've included a quick summary of the methods that are currently being used for addressing the signal extraction. So some of them are based on ICA and PCA, and they tend to group the pixels based on the correlated activity of the pixels. So if two neurons have correlated activities, they tend to group them in the same component, which is problematic because we know that in C. elegans, many of the neurons have correlated activities. They also require some sort of pre-registration. On the other hand, there are ROI techniques which are semi-supervised, and usually there's an expert that goes through the images and identifies which regions correspond to a neuron, and then based on the averages of the pixels in those regions, the signals are assigned to certain different components. More recently, there are NMF techniques that are being used for calcium extraction, and they model the data. They factorize the data matrix as two low-rank matrices. The first matrix corresponds to the spatial footprints, which determines what is the contribution of each pixel in the image to each component, and the second matrix is the temporal signals of the components. In our model, which we call deformable non-negative matrix factorization, we use NMF, but we also model the nonlinear motion in the following way. So the video in each time point, which we call YT in here, the video in each time point is written down, is represented as a transformed version of a canonical representation of the spatial footprints multiplied by the temporal signal. So the main difference between this and the traditional NMF techniques is that we have time-specific transformation parameters that transform the canonical representation into the space of the video in the Tth time point. To solve this problem, we have written down a cost function that has two parts, reconstruction, regularization, that I'm going to describe in more detail. So the reconstruction part is how well they can reconstruct the video using the parameters of the model. So YT corresponds to the vectorized version of the volumetric image. A corresponds to the canonical representation, and we have parameterized it using Gaussian functions. So there is a center for each component, and there is a shape parameter, which is a 3x3 covariance matrix of the Gaussian function. The F beta T, which is the transformation that is used to bring the canonical representation to Tth time frame, is quadratic transformation. In our applications, we observed that a quadratic transformation can provide a fairly accurate solution, and the CT is a signal for different components. The regularization is added to the cost function to ensure certain properties of the parameters. First, we want to ensure the smoothness of the trajectories and signals. So we added the difference, the square root of the difference between the cell centers in consecutive time points and the signals in consecutive time points to ensure the smoothness. We further added the determinant Jacobian, a function of the determinant of Jacobian of the transformations. So the closer the determinant of Jacobian is to 1, more realistic the transformations will end up. And that's why we added determinant -1 squared to the cost function. To optimize this cost, beta and A are updated using just gradient descent. We backpropagated the gradient using autograd in PyTorch and Atom optimizer. But the C, which is a non-negative term of the cost function, is updated using multiplicative updates. The details of the convergence of the multiplicative updates are studied in the original paper by Sung et al. To initialize, we used cell centers that are annotated by human experts, and the cell shapes were initialized using something, a covariance matrix that is close to the average size of a neuron. We studied the performance of our method using 2 different metrics. One is the correlation of the trajectory with the ground truth trajectory and the correlation of the signal with the ground truth signal. We did the comparisons against two methods. One is a 2-step method of doing a registration first and then applying non-negative matrix factorization. The other is do some sort of tracking and then do the ROI averaging. The data used for comparison is either synthetic data or semi-immobilized C. elegans videos that I'm going to describe in more detail. So the simulations are represented in the right-hand side, and the spatial model for the simulations is 3D Gaussian functions. The motion model is a quadratic transformation between time point t and t+1, and the signals are generated using a sparse binary vector convolved with an exponential kernel. So this is how the simulated data looks like. These are different time frames. Here, I'm showing the neurons in different colors so that you can see the spatial overlap, and this is the ground truth signal. And this is how different methods work. So as we see, DNMF is more successful in recovering the ground truth signals. This is a normcore NMF, which a normcore is just a registration technique that is commonly used in the field, and this is ROI tracking. And as you see, ROI is actually mixing the signals of different components. We also showed quantitatively that DNMF outperforms other methods in several conditions. I'm also going to show you a video of our simulations. So this is the video. The first row is basically the data. So this is the data. The second panel is the reconstruction, and the third is a residual. In the other rows, I'm showing the zoomed in videos for each neuron. So these are the zoomed in cells. And this is the components recovered by DNMF, and these are the ground truth components. And finally, this is a residual, which is the difference between the two. And in the last row, I'm showing in color, I'm showing the signals corresponding to, like signals are recovered by the method. And in black, I'm showing the ground truth signals. So they have a really good overlap in for the DNMF. The left panel is DNMF, and the right panel is the normcore NMF. Moreover, we studied the utility of our method in real data. Here I'm showing one time frame of the videos, which is max projected in the Z dimension. This is a video of a semi-immobilized worm's tail, and it has like around 40 neurons. And as you can see in the zoomed in videos, there's a high spatial overlap between the cells. The data set is pretty large, so a 4-minute recording is around 600 million pixels. So this is how DNMF recovers the components in the second zoomed in panel, second row of the zoomed in panels. And this is a video as I showed before. So this is data, reconstruction, zoomed in panels, and the reconstructions. This is DNMF, and on the right we have the normcore NMF, and on the bottom we have a tracking method and averaging of the pixels in a range. As you see, I'm going to just fast forward the video so that you see that the worm is actually moving and the worm is not fixed, and the tracking is doing a pretty good job. However, because of the mixing of the signals, the quality of the extracted signals is better for DNMF compared to other methods. To conclude, extracting calcium signals is important in neuroscience for understanding neural dynamics, and current approaches fail due to multi-step nature. So the NMF techniques require pre-registration, ROI techniques require tracking, and they suffer from mixing as well. We proposed a framework that combines NMF techniques with nonlinear motion modeling. And as a future work, we think that trying to solve the problem for other animal models is an important question, and also using non-parametric footprints and better motion models and optimization techniques is important. I would like to thank all the people who contributed in this work, as well as you for listening.